Thanks for joining me today on this webinar on SPADE in FCS Express. Together with me on the line is Schoenberg, who is going to take care of all your real-time questions. So feel free to navigate to the question section of the GoToMeeting window and type in any question whenever you want. I will try to answer question in real time, but if I cannot, Sean will take care of you. Okay, so this webinar, let me tell you, it's going to be recorded. So we will publish it on our website in a few days. So you will be able to see it again. Okay, so I put together an introduction with PowerPoint because I really want to give you some basics on SPADE before moving on in FCS Express 7, because as you will see, running SPADE and actually running any type of high-dimensional data analysis tools in FCS Express is really easy. It's really straightforward. So probably the main effort for you today will be to go more in depth on how SPADE works. So that's the only way in which we'll really be able to manage the SPADE results and analyze them in a critical way. So we will see how SPADE works. We will see how to create a SPADE transformation in FCS Express, how to apply to a data set, how to retrieve results, and how to deal with those results. So first of all, why we need SPADE? Well, basically because of the same reason we need any other type of high dimensional data analysis tool, because we have data set that contains more and more parameters every day and their standard way of analyzing. So multiple 2D plots, multiple nested gate, it's not really efficient. I mean, it's fine if you have clear in mind a specific population, your target population of interest, but then if you want to have a comprehensive view of what's inside your FCS file, you really need something different. And SPACE is a good example. Or for example, 2 weeks ago, we did a webinar on Disney. So those are all different approaches. If you imagine, if you think about your FCS file as a table like the one that you see here, you will see events, so in this case cells, observations, in different rows. And you will see parameters, so fluorescences, markers, antigen, in different columns. So if this is your data set, this is your multidimensional data set, what you need to do is to make it simpler. And there are several strategies, actually mainly 2 strategies in which you can make this table simpler and basically smaller. So the first one is reducing the number of dimensions. This is what you can do with dimensionality reduction tools. And the second one is reducing the number of observations. So basically reducing the number of objects you are dealing with. SPADE is one of the algorithms that allows you to do this. Because to reduce the number of observations, the main strategy is using clustering. Clustering means grouping similar events together in a way that events within the group are more similar, let's say, than events outside the group. And SPADE is just an example of clustering. Other examples are, for example, flowsums, phenograph, Kmeans, park. They are belonging to the same family. They are basically doing the same. They are trying to solve this issue with the same approach. So let's go into more details about what's clustering. Again, you can see the definition here at the top. As we mentioned before, means grouping data such that data within a group is more similar to each other than to the data in the other group. So you can see an example at right. This is a very basic example. I know it's a 2D plot with 3 population. If you do clustering, again, you are grouping similar events together. In this case, you may want to look for 3 clusters. You see the green, the red, and the black. Okay, so we are grouping similar events together. It's very interesting to define the similarity. So how you can say that two events are similar? Well, mathematically speaking, usually the metrics that you want to exploit is the distance. Okay, so if you calculate the distance between 2 points, like in this data set, in this example, if the distance is small, you can say that the two events are similar because map in the same area, in the same region of the plots. If the distance is large, then it's very likely that those two events are not similar, so they are different, right? There are different ways in which you can calculate the distance, and we will see that different algorithms basically use different metrics. But again, they are basically doing the same stuff. So the overall idea is after you group all the similar events together, the idea is that you summarize the group, like in this case, the green group, by using a summary matrix. For example, you can grab the center, okay? So basically you're getting rid of all the single events and you just summarize all of them with a unique object, which is the cluster. So this is the goal of clustering. As you can see, we are not dealing with dimension. We are not reducing the number of dimension, okay? We are reducing the number of objects. So this means that the number of dimension is unchanged. So you see in this case, instead of, for example, let's say 3,000 dots, we end up with just 3 objects, so 3 clusters, and still we are dealing with 2 dimensions. If your data files contain 20 dimensions, at the end of the clustering, you're still dealing with 20 dimensions. So each cluster still exists in the multidimensional space. This is very important to keep in mind. Clustering is not aimed to reduce the number of dimensions. Of course, you can do that, but that's a further step. Also, another things to keep in mind is that the clustering per se doesn't tell you anything about what a group is. So what's the meaning of a group? What's the biological population contains that group? So to do that, to phenotype, to characterize a cluster, again, to phenotype the center, because at the end you are dealing with the center, you will have to go through all the dimension and say, okay, this center is at 2 for the y parameters and at 0 for the x parameters. So based on my knowledge, I can say that this is single positive for the y. But you see, you can do this only because you are looking at your plots. And if you have 20 dimensions, you will still have to go through multiple plots. So regarding with that type of, let's say, problem, so the higher number of dimensions that you have, clustering it's not intended to solve that issue. So the main example of cluster, the prototypical cluster, is the K-means, which is 70 years old. It's super fast. It's very easy to explain and to understand. There are a few things to keep in mind with K-means. We will see how K-means works. K-means calculate the distances based on the Euclidean distance. So basically, it's like the straight line between 2 points. And if you look at this example, like those are the center of each cluster, okay? So basically what we want to do is to calculate the distance between each dot and the centroid, and then assign the dots to the closest centroid. So this dot is closer to this one, so it will be green. This dot is closest to the blue one, so it will be blue. And you can clearly see that in a 2D space, if you draw the distance here around the center, it's basically a circle. If you do this in a 3D space, the Euclidean distance will look like a sphere. So this is the first key point to keep in mind when dealing with K-means, again, which is just an example of clustering, but as you will see, it's the method at the basis of SPADE. So this is why I want to present you this. Since K-means distances in K-means basically generate spheres, this is why K-means is so good in finding spherical cluster. While it's also the reason why K-means it's not so efficient in clustering non-spherical population. which is usually the case in flow cytometry. So you rarely have a perfect sphere. Usually you have some, let's say, population that's spread a little bit because you have a continuum of the expression of a given marker. But we will see how SPADE can overcome this limitation. Okay, you have any question? You have any doubt so far? If yes, feel free to type in the question section. Okay. Then the other, let's say, crucial point to keep in mind with K-means is that K-means allows the user to define a priori which is the number of cluster that you want to look for. So, for example, in the plot that we saw before, this one, it was pretty clear that we want to look for 3 clusters. So this is also easy when you want to explain K-means to someone else. But it's also a kind of limitation because you usually don't know really how many cluster you have in your data set. But again, there is a solution for this as well. So overall, this is why K-means is so diffuse and so used. So let's see how K-means works. So you define the number of cluster. Basically, you type the number, like 3 in this case. Let's say that we want to cluster this data set. Again, it's a very basic data set. It's just 2 parameters, 3 very clear and very round shaped population. So let's say that we define 3. So the first step is setting up set of 3 centroids, 3 centers. Those will be the center of our cluster. And at this stage, the 3 centers are randomly set. So let's say in this 3 position, this one, and this one. Then the question that you want to answer is, which is the closest centroid? And this question needs to be answered for any single event. Once you answer this question, you assign each event to the closest centroid. Like in this case, you see all those green dots are assigned to this one, or all those red are assigned to this one, all those black are assigned to this one. And you can clearly see that, for example, when 2 clusters get in touch, even if they are spherical, the border between them is basically a straight line. Then in the next step, each centroid is moved to the center of the cluster, like this way. And then you recalculate the cluster assignment. So you are basically re-answering the question, which is the closest centroid. So you are reassigning the cluster to the new centroid. And then you move centroid and you reassign, move and reassign, move and reassign, and so on, until you stop seeing events that flip from one cluster to another one. So this is the final results. Once you reach these final results, you get rid of all the single events and you just focus on the cluster. So you see, instead of, for example, 3,000 events, we ended up with just 3 objects. And each object is basically represented by whatever you want. In this case, it's a cross, it can be a circle, a letter, a number, whatever you want. So this is what actually happening in the background. You start with a table that contains a lot of events, a lot of parameters, and you assign each event to a given cluster, like green, black, so on until the last one. And then you get rid of the single event and you just focus on the cluster. So you end up with this one. And each cluster is now defined again into the multidimensional space. You see that I'm still dealing in this case with just like 20 parameters, but you are just using a single value. Okay, this is like the summary statistic for FL1. It can be like the mean FL1 for the green cluster. This is the mean FL1 for the red cluster, or whatever you want. And you can see you can calculate summary statistic like the percentage of cells within this cluster, the number of cells within this cluster, and so on and so on and so on. Any questions? Again, feel free to type in. So SPADE. So the main point of our webinar today, it has been published in 2011, so it's now pretty well established. It was a kind of revolution in the multidimensional analysis in flow cytometry. So you see very high impact journal Science and Natural Biotechnology. SPADE, by definition, it's a clustering, of course, and it's also visualization tools. So the two main goals accomplished by SPADE are, first, clustering the data into group based on their location in the multidimensional space. And this is done by Kmeans, okay? SPADE rely on Kmeans. And then remember, you end up with a lot of cluster, but you didn't account for the multidimensionality, okay, of your data set. So if you want to characterize each cluster, you still have to look to your plots and say, this is positive for this marker, this is negative for this marker. So to overcome this limitation, SPADE, and this is why it's considered as a visualization tool, reduces the dimensionality of the cluster for visualization. So after the clustering, it reduced the dimensionality of those clusters and plot them into a single 2D plot. And the result is this. It's called SPADE Tree. So each cluster is represented as a nodes. And you see that nodes are grouped into branches, okay? And all the clusters are plotted in this single 2D plot. The advantage is that even if you have like 200 clusters, since they are plotted in this way, you can clearly see that a group of clusters that map close each other, it's likely that refer to the same biological population. So it's much easier in this way to phenotype this whole branch instead of phenotyping each of those single clusters. Okay, so SPADE is based on different steps. The first one is a density-dependent downsampling. Then SPADE run K-means clustering. You can see here at the right, actually. So this is the original data set. This is the results of the density-dependent downsampling. Then it's clustering using K-means algorithm. And then it's generating the tree. And at the end, the last step is, let's say, coloring the SPADE tree based on different statistics. So we will go through all those steps. The first one is downsampling, and this is one, I mean, together with step 3, so the graph creation, it was one of the main points when SPADE was released in 2011, one of the main advantages. So I think you have already heard about sampling. I mean, you are here to learn more on high-dimensional data analysis tools. So I'm sure you have already heard about sampling, downsampling, subsampling. Sampling is basically like keeping fewer events, and usually this is done to speed up the downstream calculation. Like you have 1 million of events, you need to run your algorithm. It's too slow to run it on 1 million of events, and so you downsample, and maybe you just sample 100,000, so 10% of the events. That's definitely speed up the calculation, but of course, you are basically looking at a subsample of your data, so you are losing information. But sometimes that's the only way you can do that, because maybe, I don't know, your computer is not fast enough. Usually sampling is made with random sampling. That has a huge limitation. So the limitation with random sampling is that you may lose rare events. So let's say that you have, again, 1 million of events. You're looking for a very rare population. If you sample like 10,000 events, so one event every 100, it will be quite likely that you will not sample your rare population at all, or that you will not sample your rare population enough to then make some robust calculation, okay? So the only way you need to, let's say, overcome this limitation is to increase the sample size, but then you will need to wait longer, or to use a different type of sampling. Also because, for example, with clustering, even if you say, I want to run SPADE on all events without downsampling, well, your clustering may be biased by very abundant population. So even if you're sampling a rare population, maybe it's so rare that the clustering will not really find it. So this is why the author of SPADE proposed to use the density-dependent downsampling, which is a really cool sampling that basically enrich in rare population. So in FCS Express, there are different ways of performing density-dependent downsampling. The one used by SPADE is called target density, or in the SPADE paper, it's just called density-dependent downsampling. And here is an example. So this is fake data set. It's just a in silico generated sample in which I've added different population with decreasing frequency. So you see I have two very dense abundant population, like 60 and 30%. So overall they cover for 90%. And then the frequency decrease like 6%, 0.6, 0.3, 0.06, and 0.01. What you see here is the distribution. Okay, it's just an histogram of the FL1 so to show the distribution of those population. So if you do random sampling, the distribution is the same. You're just lowering down the absolute number, right? So here you see that you are risking, for example, look at the blue and the red. You're really sampling like 1, 2, and 3 events. So you're risking to completely lose those population. And again, since you are maintaining, since you are maintaining the distribution, your clustering may be biased by those huge guy here at the left, so the very abundant population. If you perform the density dependent downsampling, this is what SPADE is doing. You see the distribution here at the bottom right. It's completely different than what you see on the left. So this is called target density because what you want to achieve is a kind of flat distribution. And you see indeed that those peaks are much more homogeneous in terms of frequency, right? You see in this case, the highest, so what was the highest peak, it's 30%. The second one, it's 30%, and then it's 21, and then it's 10. Like in the original data set, you had 60, 36, and 0.6. So you see this has been increased by 3.5. This has been increased by 16, okay? And so the idea is that you are sampling fewer events from the very dense area, and usually dense area are abundant population, and you're sampling more events from sparse area. And usually in flow cytometry, sparse area are rare population. What you can also do with this target density sampling, it's removing outlier. So yes, you want to increase the chance to sample rare population, but you don't want to increase the chance to sample like debris, doublets, aggregates, or whatever. So there is a parameter in the target density sampling that says below this density, do not sample at all. So that's a parameter helps in removing outliers. And this is why, for example, in this case, with the setting that I have used, the red population is basically gone. So the outlier are being removed. So this is the first step of SPADE. So the clustering will be performed on this sampled dataset, what you see on the right. Then clustering, we already know that clustering is performed with Kmeans clustering. So we already know Kmeans, it's very fast. It requires to define the number of clusters. It relies on Euclidean distance, so it works pretty well with round-shaped population. And again, this is unfortunately not the case in flow cytometry, but there is a solution. So you see in this is a real example. This is a real data set. So you see it's just FL1, FL2 from one of my files. Population here are definitely not round-shaped. So if I can see that I want to look for 3 clusters. You can see this at the right. But if I ask Kmeans to find 3 clusters, those 3 clusters are not really efficiently separating my population, identifying my population. So you see the red cluster, it's contaminated a lot. The blue cluster, it's not including all the events that I want. So the answer, the solution, it's called overclusters. So the idea is that if I cannot shape a population using a single round clusters, I can try to shape that population with a multitude of small round clusters. So the idea is to increase the number of clusters. You see 6, 12, 20, 50, whatever. And at the end, I will shape each population with a lot of small clusters. So this is the other cool things about SPADE. So using Kmeans, which is super fast, increase the number of clusters so that it still perform well with non-round population. This actually also introduced an issue, but again, we already know the solution. And the issue is that now you need to deal with a lot of clusters, like 200. And how can I know without opening all my plots that these guys here, the light green and the green, belongs to the same population? Well, we know because SPADE, it's going to create the SPADE tree. And so it's going to group clusters that refer to similar population together. So this SPADE tree, it's what's called node edge graph, and it's kind of intuitive because clusters are represented by nodes and nodes are connected by edges. And this is the way in which SPADE is working to do this. So first of all, the node edge graph generated by SPADE, which is usually referred to as the SPADE tree, is 2D representation of the cluster relationships. So the idea is that if two clusters are close each other into the high dimensional space, so let's take this example. So the blue one is close to the red one. You represent each of them as a nodes, and then you connect them. So you see those are the three clusters that you see here, okay? And overall, this is the shape, okay, of the original data set. Of course, it's a two-dimensional representation of the original shape. So this is what actually happened in the background. So you calculate the clusters, you calculate the centroid, and then you work with your centroid, right? The distance between cluster, so between centroid, it's calculated as Euclidean distance in the multidimensional space. Then each cluster is represented as a node, and nodes are connected together by edges in a way that each cluster is connected to its closest neighbor. And this is the spanning tree. So starting from the left, you have your clusters. You calculate the distance between each cluster and all the other clusters. For example, the distance between A and E, it's 6. The distance between A and C, it's 7. A and B, it's 1. We should also calculate A and D, A and F, of course. And then you get rid of all the distances except the smaller one. So for example, you see F, it's connected to B, so B is the closest neighbor. I want to keep this edge. And this is why in the final tree, F, it's connected to B. A, it's connected with E with a huge distance, C with a huge distance, D even bigger, and B, it's the closest neighbor. So I want to keep the edge between A and B. So at the end, you see that each node is connected with the closest neighbor. It's very important to keep in mind at this stage that the overall shape, so the aesthetic of the tree, again, it's just aesthetic. It doesn't matter. So the only thing that matters here is the way in which nodes are connected. So whether or not a node is connected to another one. And you can clearly see this at the bottom. So this representation, so you see A, B, C, D, E, and then F, it's linked to B, right? This representation, it's the same representation that you have at the right. So A, B, C, D, E, and F connected to B. So the way in which you lay out all those nodes into the plot is just aesthetics. Only whether or not a node is connected matters. Then last step is calculating all the statistic and actually upsampling. So remember, in the first step here, we basically downsample, so we remove a lot of events because we wanted to achieve a flat distribution. So what happened to the unsampled events? Well, once you have the SPADE tree, you're going to put back all those unsampled events. Basically, for each of those unsampled events, you are answering the question, which is the closest centroid, and basically which is the nodes to which that event belonged, and you assign that unsampled event to the nodes. So why this? Because at the end, if you want to calculate, for example, the MFI for a given parameters like FITC MFI of a single cluster, you don't want to just calculate the MFI based on the sampled data set. You want to really calculate the MFI with all the events in that cluster, so to have a more robust statistics. And then you can play with, as you will see, you can play with the SPADE tree. You can color each node based on a statistic. For example, you see here in our data set we have marker 1, marker 2. If you color based on marker 1 MFI, you will see some red nodes, so you can conclude that those are highly expressing marker 1. If you do the same with marker 2, you will find other nodes. And by comparing these different SPADE tree, I mean the same SPADE tree colored by different parameters, you can conclude that this branch is double positive, this is single positive for marker 2, this is single positive for marker 1, and this is double negative. Okay? Do you have any question? Do you have any doubt so far? Okay, no question. So what you see here is basically the final results. You have all your nodes. You see there are a lot of nodes. You have branches, and then you can color each nodes based on a given statistic. For example, in this case, the CD3 FITC arithmetic mean. So I can conclude, for example, this branch contains CD3 positive cells. But I want to also show you that, for example, I have other nodes that are CD3 positive, that are red, and thus probably contain CD3 expressing cells. So you may ask yourself why they are not all grouped together in the same branch. Well, because we are just looking at CD3, and SPADE, to create this tree, looked at all the parameters. So it's true, those nodes are CD3 positive, those nodes are CD3 positive, but they are different in a different position of the tree. And this is because they are definitely different with regard of other markers. For example, they can be like CD4 positive, and the other one can be CD8 positive, and the other one can be double negative and double positive, and so on. Okay, let's move to FCS Express. Let me just open a blank page. I will not go into any basic stuff, so we'll have many other webinars that can present you the basic functionality. So I will go straight to create a t-SNE transformation. So let me just open one single file. We will do something more advanced later on. So here's my file. I'm going to drag and drop and to open a density plot. And let's say that I want to focus on something very simple here, like a lymphocyte population. Okay, this is my cells population. And I want to calculate to run SPADE on this population of interest. So when you want to create a SPADE transformation, just go to Tools, Transformation, open this drop-down menu and select the transformation that you want. And today we will, of course, select SPADE. Let me increase this a lot. I want to show you the entire window. We will go through all those options one by one. So the first step is, which is the gate of interest. So which is the population that you want to run SPADE on? I mean, it's not mandatory, of course. You can say I want to run SPADE on all events. That's fine, but usually you won't. You won't because maybe you want to clean up by removing dead cells, by removing doublets, by removing debris, or maybe you want to focus on a specific population, for example, lymphocyte, or maybe T cells, B cells, whatever. And you can do this very easily in FCS Express. Just select any gate you want from this drop-down menu. Then you need to select the file. In this case, the file it's automatically selected, so the software is smart enough to understand that I'm dealing with this file here. But if you're dealing with multiple files and you want to run SPADE specifically on one file, just click on this ellipse and grab the file from your computer. Once you select the file, you will the software will retrieve the parameter list. And this allows you to select which parameters you want to include in the SPADE transformation. Again, why you need to select the parameters. It's not mandatory, but usually you want. Why? Because you don't want to include parameters that are not informative. For example, you're not going to include the time. The time is useless in this case. The time is not giving you any useful information about how your sample is distributed along different parameters. So it's not giving you any advantage in terms of separating subset. So it's just introducing noise. So when you need to define which parameter to include, always ask you the following question. So is this parameter giving me some advantages? So it's informative. And of course, there is not like a universal answer. For example, what about forward scatter, say scatter? What about CD45? What about whatever? So usually when I present this, there is a question that always came up. So the question is, let's say that I'm interested in a specific population. Like in this case, it's lymphocyte, but it can be, I don't know, CD3 positive cells. Okay, the question is, do I need to include the CD3 positive cell, sorry, the CD3 marker? Or in my example here, do I need to include forward scatter? Do I need to include side scatter? So the question in this case is the same as before. So let's grab this as an example. So I'm looking at lymphocyte. If you think that the forward scatter variability within the lymphocyte gate, so the variance of the forward scatter is biologically relevant, then include the forward scatter. If you think that this variability, okay, so you see I have some cells at 65,000, some cells at 131,000. If you think that this variability, it's not because of the biology. So it's not because some subset some subset have an higher forward scatter, some other subset have lower forward scatter. So if it's not based on biology and it's just because of the instrument variability, in that case, it means that forward scatter is not informative. Okay, so you can skip it. And the same is true if you're creating a gate on CD3 positive cells. So the question is, do you think that the CD3 expression level within the CD3 positive cells, it's biologically relevant? If you say yes, because I know that there are CD3 bright, CD3 low, CD3 intermediate, and those can correlate with different subsets, in that case, include the CD3 because you want to, you want to keep that information in your calculation. If you say no, I think that within the CD3 positive cells, they are all expressing the same amount of CD3, and that's just technical variability. In that case, you will not check the CD3. Then the other crucial point is for each of those parameters, you need to define a scale. This is really important. And I actually have a slide here. So you may already know that in flow cytometry, the variance increase with the increasing of the intensity. So this means that if you look at the raw data inside your FCS file, and if you plot, for example, any fluorescence marker, like in this case, you see here I have a CD3 FITC, and if you look at the raw data, which means the linear scale, you can clearly see that the positive peak is broader than the negative. Okay, so you see the variance, it's increasing with the increase of the intensity. And if you have a peak here, it will be even broader. Okay, this is how, this is how the instruments are defined. So this is also why you basically always apply a log scale, a B exponential, an hyperlog scale every time that you plot a fluorescence marker. So every time you're doing this every day, you see in this case, it's the same data set as above, but with the exponential scale, okay? So what you're doing is, as you can see in the title, you are making the variance more comparable between the negative and the peak. Sorry, between the negative and the positive peaks. Another way to look at this is that if you look at the raw data, you can conclude that distances at higher intensity are less important than distances at lower intensity. And remember, K-means, but also many other clustering and dimensionality reduction tools rely on the distance to evaluate as a matrix to evaluate the similarity. So you can clearly see that if you're working with this type of data and then you compare distances between within the positive population and the distances that you have calculated within the negative population, you're comparing apple and pears. While if you apply a scale, you make those distances more homogeneous. You can see the green bar, the green marker and the orange marker. Okay, now you are comparing apple with apple. So the only difference between what you're doing every day on your histogram or on your 2D plots and what you're going to do now with transformation is that every day on your plots, what you're doing is just aesthetics. Okay, you're not changing the actual value. So if you have a cell here at 1000, that cell will still be at 1000. So you're not manipulating the value. In this case, since SPADE needs to perform some mathematical calculation, you are actually converting the value based on the scale. So for example, 10,000 in log scale, it's 4. Okay, 1000 it's 3, and you can clearly see that the distance that here in linear scale it's not homogeneous becomes homogeneous after applying the log scale. I mean, log scale is just an example. I would actually suggest to not use log scale for fluorescences because it doesn't handle negative value, but you get the point. I mean, nowadays there are all the hybrid scale like hyperlog, b exponential. So but they are doing the same stuff. So they are stabilizing the variance. So let me go back here. How can I define which is the right scale for a given parameters? Well, what I usually suggest is just look at your data. So what I usually do is opening a plot gated on the population of interest. And you see here with the multi-plots, I can create the multi-histogram. So now I'm interested in these parameters. And this is the multi-histogram. So what you need to look at here is whether or not your peak, positive and negative, are, let's say, comparable. I mean, not in terms of frequency, comparable in terms of variability, okay, in terms of shapes. And you see, I cannot see anything really bad here. Remember, this is bad, okay? Because there is a huge difference in terms of spread between the negative and the positive, while in this case, they are pretty comparable. And if you click on one of those histogram and you open the formatting axis here, you see by default, and in this case, those are scaled with b exponential automatic setting. So if you're fine with this, they are all scaled with b exponential automatic setting. You can use the same here. If you see something strange and you want to play and change the scale here, then feel free to go here and say, for example, the CD4, I want to still use b exponential, but maybe I want to play with the below 0, or I want to play with a different method. So in a way to make the two peaks more homogeneous, but in this case, it's fine. Then the sampling option, of course, target density. This is basically the only thing you can select with SPADE. I mean, you can change it, but if you want to run the original SPADE, you need to select target density. I'm not going to much detail here because there is a lot of math underneath. If you are interested, just shoot me an email, or you can also have a look at the manual. Just very briefly, you see this is the target density. So this is the flat density that you want to achieve along all your data set, and this is the density below which you don't want to sample. So this is the minimum density, also called threshold density. So below 0.01 you will not sample, but keep in mind this is not the frequency. This 0.01 doesn't mean 0.01%. This doesn't mean 0.2%. This is the density. It's not a percentage. So again, if you have any doubt, just shoot us an email. But this is anyway the place in which you can play with this. Transformation option, number of cluster. This is the number of centroid. Remember, you need to overcluster. That's at the basis of SPADE. If you're expecting like 10 population, 20 population, you will not type 10. You will not type 20. This is like 100, 200, 300, whatever. Minimum percent set per cluster. Again, it's some kind of advanced setting. This is just to avoid that during the while Kmeans it's running, you are not creating like empty clusters. Maximum number of iteration is just to stop Kmeans in case it didn't reach the convergence, but usually it doesn't happen. I mean, usually it converges in flow cytometry because we are not really dealing with a weird scenario. Number of clustering attempts, this is pretty cool. So remember, the first step of K-means, it's random initialization of the centroid. So if you want to increase the chance to have better results, you can try different random sets, because of course, I mean, the random set of centroid that you select at the beginning, it's somehow influencing the K-means clustering, so the results. So if you try different centroid, different set of centroid, then the software will pick up the best results. So feel free to increase this to any number you want. Of course, if you increase the number of attempts, you will need to wait longer. Like if you run one attempt, maybe it's run in 2 seconds. If you run 10 attempts, it will run in 20 seconds. But it may work. This is another very specific. So if in case K-means didn't converge, do you still want to keep the data, or you just want to see an empty plot? So this is up to you. Graphing layout, we will see both. So those are just two aesthetical layout that you can apply to SPADE. And last but not least, the seed. Again, it's a way to ensure the reproducibility. Since the first step, it's a random initialization. Ideally speaking, if you run SPADE twice on the same dataset, you should end up with two different results, which is not really acceptable nowadays. So this is a trick that informaticians use to ensure that if you run SPADE twice on the same dataset with the same setting, you end up with the same results. Okay, so let's say that this is our SPADE transformation. Let me create a little bit of space here. You can also dock it. I want to show you. See, I'm dragging and dropping the SPADE transformation dialog here, so I can keep it on my side. If you want to access SPADE Tree, you go here in the Insert tab and you open the plate heatmap. So now it's going to be empty. It's going to telling me you don't have any parameter compatible. I say yes. So now it's empty. And what I'm going to do is to drag and drop the SPADE transformation on top of this map. And now you see I have my SPADE tree. So you see 100 nodes in my plots. This is the arch layout. It's also called U-shape because you see it's a U-shape from which you see different branches that grow up here. I also like this one, the radio, which is basically, it starts from the center and then it's all the branches goes out, but you see it's the same, so it's just aesthetic. So if you look at all those branches, like you see this here, you have 1, 2, and then you have a little branch and a little double branch. You will see the same if you go with the arch. So you see here, this is 1, 2, a little branch and a double branch. So it's just aesthetic. Again, what matters here is whether or not two nodes are connected by a hedge. Okay, so now that we know how to apply SPADE, let me open, let's say, a nicer example here. It's the same data set, just with more events. You see I've created the gates on lymphocyte and monocyte actually. This is the standard way of analyzing data, so multiple plots, so multiple gates, multiple combination of parameters. And this is actually what we're dealing with tonight. So, sorry, today. It's the SPADE tree. You see, this is my SPADE tree. I want to spend few seconds here to show you how this plot can be customized. So first of all, you see that each node it's colored with a color, and you can define which color to use. So which metrics to base the color on. And you can see here on the left. So you see this is the median of the PE channel. So when you see red, it means in this case it's high. When you see green, it's low. So I can clearly see, for example, that those three nodes are red, so are positive. Those nodes are positive. Those nodes are positive. If you click here, you can change the parameters. So for example, where are CD8? I can click on CD8 and I can clearly see that I see CD8 in this branch. I can clearly see CD8 on this branch. You can also change the statistic. You see, this is the median. If you click on the SPADE tree and you go to format overlay, you see here you have all the statistics that you want, like median, geometric mean, arithmetic mean, percentile, and this is the parameter. This is the same parameter that you can change by acting on the y-axis. Another cool thing that you can do is to make the radius of each node proportional to a given statistic. And you can define this here. So let's just play, let's say CD8 median, so the same that we're using for the coloring. And you can already appreciate that now nodes that contains events with an high CD8 expression are red, but are also big, okay? But cool things, for example, is to use number of events. And now I can clearly see which nodes are red, so express CD8, and which node contains more events. So for example, these two guys here contains more events than these little guys. Now, one question for you. You see that the CD8, it's again distributed in mainly in two different branches, right? So why? Again, for the same reason we saw before. So because yes, they are all CD8 positive, but they differ based on something else that we don't know yet, right? So what you can do to explore this SPADE tree, the first strategy that I want to mention is backgating. So as you can see here, I have created a bunch of different color dot plot. You don't need to create them one by one. It's very easy in FCS Express. You can just insert a color dot plot and select it, and you see here you have multiple plots. So if you click on n by n plots, you will be able to create what you see here at the right. So no need to create them one by one. Now what I'm going to do is to create gates on my SPADE tree. So I'm going to gating, I'm selecting well gates, I'm draw a gate here. For example, I want this branch. This is CD8 branch 1. I click on close and then I create here. This is CD8 branch 2. Sorry. Okay, they are both CD8, but they are different. And I know that they are different, even if I don't know why, but I know that they are different because they are in different branches. I can keep coloring the gate by right click and show gates. You see, I can activate them, or I can also select all. So I can keep looking at these gates here. And now you can see the backgating on the right. And you can see, for example, let's have a look here, this one. So on the XCDA, they are both positive. We already know that. And you see that they differ in terms of PE. So the red one, it's positive. The green one, it's negative. Let me also do this. I want to make this gate more visible. I will click on emphasize. Okay, you see, Now they are pretty more visible. And this also allows me to show you something. So let's focus now. So again, this is the first strategy. You see, you create a gates on the SPADE tree and you backgate on standard color dot plot. I want to show you this. Let me get rid of the green one. Let's have a look at these red gates that we have done by including these 4 events. Sorry, these 4 nodes. You see that, yes, it's a kind of homogeneous, but actually it also includes some, let's say, heterogeneity. Like you see, most of those cells, I mean, they're all CD8 positive, but they include also some CD4, right? So what I can do is right click, edit gate, and then explore each of those nodes by just clicking. Okay, now this is one node. It's purely CD8, purely CD8. I'm looking at these plots, and this is CD8. You see, these nodes contains both CD4 dim and CD4 negative. So there is nothing I can do now with this SPADE tree to further discriminate, to separate these 2 populations. So when you see this, so when you see that the nodes include populations that are, I mean, clearly not similar, okay, it's because you define number of nodes which is too low. And in this case, indeed, I intentionally set my number of 250, okay? So this means that the number of nodes, it's not enough to separate this. So if this happens, just go here and increase it. But again, we already know that 15, it's not enough. What I want to show you is that, let me get rid of this plot, is that you can also do the opposite. So you can create a gates on, for example, the CD4 population, and then drag and drop it on your SPADE tree to see where those CD4 are. So now I know that the CD4 are here at the bottom right. This is actually more or less what you can do. Let me go back. Okay, what you can do if you just display, sorry, if you use the CD4 to color your nodes. So you see, we know that they are here. Make sense? Do you have any questions? Okay, so I see a question about how to deal with multiple data file. We'll see this in 2 minutes. Yes. So this is the first strategy, so using backgating. The second strategy is using histogram. And again, this is pretty cool because in FCS Express it's really easy. Let me just create a gate as we did before. So for example, this one, okay? This was CD8 branch, branch 1. I want to apply this gate to my histogram. And now what I'm looking at is the CD3 expression. But what I want to do is to look at the expression of this gate through all the markers. And look at this, right click, add parameters overlay, click all the parameters that you want to display as a new overlay. I will switch to stacked and I will click OK. So this is the phenotype of my population, of my CD8 branch 1 gate. You see CD3 positive, the PE, it's a little bit positive and negative, sorry, negative and positive. It's definitely CD45 positive. The CD4, we already know that most of them are negative, but there are few contaminants here because we are using not enough nodes. And last but not least, CD8, it's definitely positive. Okay, this is another way. Of course, you can play with this and you can do any sort of embellishment, like you can select all the overlay, you can fill, you can add transparency, you can smooth, you can do a lot of stuff. But this is just aesthetics. So the main point is a clear picture, a clear phenotype. And to answer that question about how you can deal with multiple files, well, as it happens with most of algorithms that rely on distances, you cannot run like SPADE on different files separately, because you will end up with different SPADE trees. So it would be very hard to compare them. So what you need to do is to first merge all your files together and then run SPADE on the merged file. So we don't have time to see how you can merge file in FCS Express. We have a webinar for that, but it's very easy. It's just like 10 seconds. But once you've done that, in the merged file, you can still separate each single FCS file. So this is what I've done here. This file, let me first show you this one. Like you see, this is my file. It's for scatter size scatter, but this is actually the merge of 8 different files, and I can discriminate them because when you merge file in FCS Express, a file identifier, a new parameter, as you see here at the bottom of the list, it's created. So what you see here is file 1, then file 2, file 3, file 4, file 5, till file 8. Another nice representation in FCS Express is this one. It's the heat map. It's the plate. It's the same heat map that we can use to plot SPADE. And in this representation, if you merge 8 files, like in my case, basically they are represented as wells in a, let's say, ideal A to L plate. You see, each well is a gate. It's a file. So the idea is that now you can, for example, create a gate on either the file identifier or on your heat map. So, sorry, let's say that this is file 4, okay? And now your gates are available in the gate hierarchy. You see the file gate, which is this one, the file 4 gate, which is this one, and you can use them. You can drag and drop file 4, for example, on this plot and look at file 4 only, or on the SPADE tree. And for example, you can see that file 4, it's missing this population. So the node is empty and the node is empty. And this may be something cool that you want to explore. Well, it's also clear here, we are coloring based on CD19. So this is a population of CD19, and you can indeed see that CD19 are quite rare here. So that makes sense. You can also use these gates. And if you use the gates on the file identifier, it's even easier to scroll through files. You see here. It's very, very easy. So this is the flexibility that you have with FCS Express. And then I'm not going to more details because we don't have time. But again, this is just another example in which I have different SPADE tree. I open different representation of my merged file. I get each heat map based on a gate, like this is depicting the percentage of CD4, this the percentage of CD8. You can also grab from this heat map the actual numbers, the actual value. You can plot in FCS Express using box and whisker. So there are really tons of stuff that you can do downstream. Okay, I don't want to waste too much time. So I really like this topic, so I can keep speaking really a lot. I just want to conclude. SPADE, it's really fast. It's really smart because of the density dependent downsampling, because of the SPADE tree representation. The disadvantages are mainly, let's say, derived by Kmeans. So Kmeans is faster, but it's not performing well on non-spherical population. But again, you can overcome this limitation with overclustering. K-means is based on Euclidean distance, and also the upsampling is based on the Euclidean distance. Euclidean distance is just one of the many ways in which you can calculate the distances. So it may or may not be the best solution, but SPADE, this is what SPADE is done. And last but not least, I want to really stress that when you're dealing with multiple files, you're merging file, you're running SPADE. Actually, also when you're running any other type of algorithm, the standardization is really important. So if you merge file that has been acquired in different run, or on different instrument, or on using different antibody, it would be very hard to get meaningful results unless you took care of the standardization. Because again, if you're dealing with file that differs because of the technical variability, SPADE doesn't know that that's technical variability, so it will map that technical variability as well. So if you can reduce the technical variability, then you can really rely on what SPADE it's spitting out. Okay, so let me just go back to my first page, and if you have any question, that's the perfect time to typing in. Okay, I can see a question here. Also, basically, what we can learn from SPADE instead of like t-SNE and vice versa. I mean, it's hard to say, those are completely different approaches. So in t-SNE, you're still dealing with a single events value, and you're just reducing the number of dimensions. You still have the visualization, so you're still, let's say, grouping graphically events together in the t-SNE map, but you're not creating groups. So you are the one that draw the gates on the t-SNE islands, right? While in this case, the software, I mean, SPADE is drawing, let's call them gates, but it's drawing clustering. It's true that you're still creating gates to group clusters, that's true, but the, let's say, the border between each cluster, it's calculated by the software. And just to conclude, because this is a great question actually, there are people that, for example, are running t-SNE and SPADE in parallel, and they use t-SNE to just visualize the clustering, to just, let's say, double check that SPADE performed well. And there are also other people, and this is a kind of interesting approach, that do not really run SPADE on the data set. They run SPADE on t-SNE. So they use SPADE to cluster the results generated by t-SNE. So you see, it's a really interesting topic, this one. I think those great tool, it's true that they are around for a while, but I think that in flow cytometry, they are still, let's say, kind of new. I mean, t-SNE, FlowSum, t-SNE, SPADE, UMAP. So I think we are learning every day how to use those data sets. And again, I also want to mention that there is nothing like the best practice, because on some data set you may get better result with SPADE, and some other data set you can get better result with t-SNE. So it's really learning from doing. Okay. I don't see any additional questions, so I really want to say great thanks. I'm sorry I was a little bit late, but I think it was useful. Okay, I see one more. Okay, other questions are coming. I will probably answer that offline. So many thanks to all of you for attending, and just look at our webinars page to look at new webinars. Many thanks to all of you.